Hello, everyone. In this video, we're going to see how fatty acids are made. So when we broke them down, they always were, they had a thioester with coenzyme A, and breakdown we saw happens in mitochondria, and we use NAD and FAD because we need to pick up these electrons, right, from fatty acids. That's an excellent way. I mean, this is an excellent reason why fatty acid breakdown gives up so much ADP, because all these electrons can go on then onto the electron transport chain, and you can get ADP from ATP synthase. In synthesis of fatty acids, some steps are really in reverse, especially some of the chemistry. That's done when we make the fatty acids and we reduce it, but as opposed to here that we oxidize it. But there are some differences. So instead of thioesters with coenzyme A, we have thioesters with something that is like coenzyme A, called acyl carrier protein. So we'll see how that works out, but it's still a thioester forming chemistry there. Now, instead of mitochondria, which is where the breakdown happens, here all the synthesis happens in the cytoplasm. And instead of getting electrons out of the fatty acids here, of course, in the synthesis, we have to put them in, and we have to fully reduce them. So what's the reducing power is NADPH, which, as I said in a previous video, is a major sink of NADPH, of reducing power in the cell, especially in cells that have to do a lot of biosynthesis for new membranes. This is where they spend a lot of their NADPH. Just so we are all on the same page again, remind everyone what's an acyl group. It's really some prosthetic group here, a long carbon chain with a carbonyl at the very end. All right, so acyl carrier protein. We don't have a coenzyme A in the synthesis part, but the chemistry, again, the acyl part is going to be make a sulfhydryl with this thiol at this point, which is one end of a small cofactor called phosphopantothane, and phosphopantothane itself is bound to a protein called the acyl carrier protein. So you have to imagine a giant protein at this end. It has a cofactor phosphopantothane, and then the thiol group here is the business end where the acyl group will attach, and the whole thing is called acyl carrier protein, which is a component of fatty acid synthase. And that says the same thing, all these arrows. All right, so when we make lipids or fatty acids, we have to solve three major problems. So every time you stitch, carbons together, you have, this is not thermodynamically favorable. Things want to be broken up as opposed to being put together, thermodynamically speaking. So we have to displace somehow the equilibrium towards carbon-carbon bond formation. And we'll see one way that contributes to that. And of course, as I just already mentioned, the fatty acid side chain is very reduced, so we have to take oxidized carbons really in the form of keto or carbonyl carbons all the way to hydrocarbon state, which is methylenes. Right? And not only that, but we know that some of the chains of this fatty acid is quite long. In fact, fatty acid synthase makes usually 16, sometimes 18 carbons in a row. So first things first, so let's make the building block. The building block is not acetyl-CoA, it's actually malonyl-CoA, which has 3 carbons. And we'll see why that is the case, or needs to be the case. So the enzyme that provides this building block to fatty acid synthase is called acetyl-CoA carboxylase. So it does exactly what the name says it does. It takes acetyl-CoA and carboxylases. There is a carbonate. And as with every carboxylation, the problems that need to be solved here by acetyl-CoA carboxylase is, what's the species, what's the source of CO2? If it's carbonate, it has to be activated because it's a poor electrophile. And we have to stabilize the carbanion that will form at some points, which will attack the CO2 to extend by 1 carbon to be carboxylated. So we have seen the solutions before, and it's exactly the same solution here. Acetyl-CoA carboxylase is a biotin enzyme. So first, the carbonate gets activated with ATP. This is where you spend your ATP zone here to activate carbonate, to activate CO2, which is fairly inert. And once you do that, and you have carbonyl phosphate, then biotin is used to keep it in place. Okay, that's the purpose of the cofactor here, so that it can be attacked by the carbanion, the acetyl-CoA carbanion. And we've seen how that gets stabilized. You're going to see it in the next slide too. I'm just going to tell you now again, that's a keto-enol tautomerization that will stabilize the carbanion. But anyway, when the carbanion is formed, then it can attack CO2 and you get malonyl-CoA and you generate biotin on the enzyme. So the mechanism is no different from what we have seen for other carboxylases. Except, of course, in this case, it uses acetyl-CoA as the substrate. So, and that's the stabilization of the carbanion of acetyl-CoA. Right. So this is the activation sort of step of the building block. That's malonyl-CoA. It's already in the it's got 3 carbons in the thioester with CoA, and it's ready to be used now by fatty acid synthase. So fatty acid synthase, what it does is to first is to take acetyl-CoA also and acetylate itself. So instead of having a sulfhydryl with coenzyme A, a thioester bond with coenzyme A, you have a thioester with a thiol on the enzyme. Okay, now as a result of this reaction, number 2. So you have an acetylated enzyme now. The acetylated enzyme will react with malonyl-CoA, which is not, as you see, it's not free malonyl-CoA. It has already malonylated the enzyme. So wait a minute here. So malonyl-CoA comes in, okay, on the enzyme, just as acetyl-CoA came in, and acetylated enzyme itself, malonyl-CoA comes in. Also, it malonylates the enzyme, so you just swap sulfhydryl thioester bonds there. So in both cases, you release 2 coenzyme A's. So what you end up with now, and you're ready to stitch them together, is acetyl and a malonyl group on the enzyme. Okay, so that's 2 carbons, that's 3 carbons. And they do come together. So you have 2 plus 3. Malonyl is the building block, okay, and you make now 4, okay, because you've lost 1. So 2 plus 3 it doesn't give you 5; it gives you 4 plus 1, which is CO2 that goes away. So first you carboxylate, so you have an input of 1 carbon, then you lose it every time you extend by 2. So what has happened now from this reaction number 3 is that you took the 2 carbons that were on the acetyl group, and you extended it by 2 more carbons. Right? But in the process, you lost 1. How did you extend it? Because you brought in malonyl-CoA. That's the 3 carbon unit that's used as building block. Why do you do that? I mean, why do you first put 1 carbon in to make malonyl-CoA and then you take it off malonyl? And you only extend by 2. So what's the purpose of this this reaction? That doesn't make any sense. Why do you put it in one place and then they remove it the next? Well, the enzyme fatty acid synthase has to solve 3 major problems. One is to displace the equilibrium towards carbon-carbon bond synthesis. And notice here how you break a bond actually, and you make it irreversible, and you release something that can go away, and that is how you drive biosynthesis towards the extension part. So one of the products goes away, it disappears, and you also break a carbon-carbon bond so that this can happen, and this is what makes it irreversible and thermodynamically feasible. This is the reaction, as you can see. So that's the malonyl on the enzyme. That's the thioester on the enzyme. That's the acetyl thioester on the enzyme again, right? So CO2 leaves, and then the carbanion forming here on the malonyl will attack the acetyl on the enzyme, and it will be joined, okay? And then you have, and then you are done. I mean, then you keep going. So you have extended by 2. Now it's time to extend with 2 more, and you keep doing this. We'll see how this gets happen. But before you can extend, look at the carbons that you have that you're left with. Okay, so you have This carbon here is in the the oxidation state is a carbonyl carbon. If you want to make it longer and longer and look like a fatty acid, you have to reduce it. Okay? So we have to reduce the beta keto carbon down all the way to hydrocarbon state, to methylenes. So this part is happened actually exactly in reverse from what we saw in the fatty acid breakdown. So you have the 3 keto acyl group, and you bring in what you do. What do you do in this case? Well, first of all, you take the keto and you convert to hydroxy. And once you have the hydroxy, you can dehydrate. Okay? And once you dehydrate and you have a double bond, then you can reduce it further all the way to the methylene state. We actually did exactly the inverse when we're going into to beta carbon oxidation. So we started with something like this before when we're breaking fatty acids. And the way to break them down is first to oxidize them and make the double bond, which can then be hydrated, okay? And then once it gets hydrated, you can further oxidize the hydroxycarbon to a ketocarbon. Right? And then you can break it with thiolase at the very end. So this is actually exactly the reverse. But notice how the reducing power is used enormously here. You have to spend 2 NADPHs for every 2 carbons that you add onto the fatty acid. That explains why fatty acid synthesis is such a major sink of reducing power, the NADPHs that you made so copiously in the pentose phosphate pathway, for example. And you keep doing this. So this is, once you do this once, then you keep doing all these reactions, 3 through 6, again and again and again until you get to the chain that you want, 16 or 18 carbons. And finally, you have to release, right? So that's the idea. But how do you keep doing this again and again and again in a very efficient assembly line type of thing, like as if you're making one car after another, after another, after another, which is every 2 carbon unit that gets added from malonyl, you have to redo it again and again and again and reduce it and so on and so forth. Well, all these reactions that you see is actually one giant complex, one big protein that does all this that has all these activities. So it is an assembly line, and you see the various numbers here, or wherever all these reactions are, and you can actually see the central role of the acyl carrier protein that shuttles them to wherever they need to go to be processed, right? So it's a big macromolecular complex, fatty acid that is, and it was a major triumph when the crystal structure was actually solved of this giant complex. All right, so the overall reaction now looks like this. You have acetyl-CoA plus 7 malonyl-CoAs. So malonyl-CoAs keep coming in, and in the end, what you have, let's say if you keep doing this, enough times you get palmitoyl-CoA and you consume a ton of NADPHs. And for every malonyl-CoA that comes in, a CO2 comes out. If you include the acetyl-CoA carboxylase reaction, ACC, then this is the overall scheme. And there you also include ATP, right? So because you have to activate a carbonate to carboxylate acetyl-CoA to make malonyl-CoA that you see there. And there you have it with the whole biosynthesis, the scheme, which we have seen many times already. Okay, what do? You start with something small to make something big, and in the process, you use ATP and NADPHs, energy and reducing power. So that's a constant thing. We saw in the first part of the class how you make ATP and how you get reducing power through pentose phosphate pathway, and this is where you put it to use, right? But we're not done yet, because this is so acetyl-CoA. I told you that fatty acid synthase and ACC1 are actually cytoplasmic enzymes. They work in the cytosol. How do you get all this acetyl-CoA to come out of the mitochondria? Remember, this is where you break down fatty acids, or you bring in, pyruvate to enter the TCA cycle as acetyl-CoA. It's all in the mitochondria. It needs to come out to engage with this system, okay? And acetyl-CoA carboxylase actually, especially. So how does that happen? Well, thankfully, the acetyl-CoA that's in the mitochondria, as we saw with citrate synthase, we make citrate. It's actually citrate that can come out. And once citrate, there is a specific shuttle that brings in citrate out of the mitochondria and pyruvate in the mitochondria. And that's yet one more shuttle here. We're not going to go through this in detail, but suffice to say that the source of acetyl-CoA for acetyl-CoA carboxylase is citrate. Citrate is broken down. I mean, you release the citrate by ATP citrate lyase, and which generates oxaloacetate, and we have seen how that gets back into the mitochondria. And now this acetyl-CoA can be used for fatty acid synthesis. So this scheme that we just described, the first reaction is acetyl-CoA carboxylase, and all these others, the 2 to 7 is actually fatty acid synthase. It will generate even carbon, saturated fatty acids, right? So this is a standard, fully saturated, all protons on the carbons, and it's even numbers. Why is it even? Because you add 2 at a time, net. You add malonyl-CoA, but you lose a CO2 every cycle, so the net gain is 2 carbons. So, and it will stop at about carbon 16 or carbon 18, mostly carbon 16, some stearate carbon 18 is also made. How do you get all these extra ones? Right? Well, if you start at the very first step, instead of carboxylating acetyl-CoA to get malonyl-CoA, well, then you start with one bigger, propionyl-CoA. You carboxylate, so you have a 4-carbon unit here. Okay, so when you feed that into just one cycle in the fatty acid synthase, you're going to lose 1. 4 minus 1, that will be 3, a net gain of 3. And then you keep adding 2 to that, but in the end, you're going to have an odd number fatty acid being made. So that's pretty straightforward. You just start with something that's 1 carbon extra. In humans, we don't really have to worry about the unsaturation because we get them mostly from our diet. Okay? So most polyunsaturated fatty acids must be present in the diet, so we don't actually make them. Now, how important is fatty acid synthesis? And how is it regulated? Well, it's regulated very tightly, especially at the acetyl-CoA carboxylase step. It's a very actively regulated enzyme. I have to say that in my lab, we work a lot on acetyl-CoA carboxylase and fatty acid synthase. We know they're regulated at the level of gene expression, how much you make in the cell. But this is another way that's regulated, which we have seen already now. It's a post-translational modification by phosphorylation. We have even seen the kinase that will put this decorate acetyl-CoA carboxylase with a phosphate. It's AMP kinase, AMP-dependent kinase. So what will happen if acetyl-CoA carboxylase is phosphorylated? It will be turned off. Okay, let's think about this now. Phosphorylation of ACC will turn it off. Who puts the phosphorylation? This kinase. And how is that regulated? By AMP. That's why they call it AMP-dependent kinase. If you have a lot of AMP, the kinase will be on, ACC and fatty acid synthesis will be off. Okay, so AMP on, AMP high, ACC off. Is that a good outcome? Let's think about it. If AMP is high, it means your energy status is low. Okay, it's a sentinel metabolite for low energy status. So you need to go on, make ATP. Don't waste your time making lipids. So shut it down and don't make lipids. So that's the idea. So it makes perfect sense, okay, to be regulated in this way. It turns, how important is it? Well, it turns out it has some very profound implications. There is a hormone called leptin. And leptin is going to be sent by our brains, the hypothalamus. It's going to find some adrenergic receptors, and it's going to bind to them. And actually, it's going to find its own receptor, the OB receptor, and together with other signals from adrenergic receptors, they will activate AMP kinase. So if leptin is present, the message is from the brain is leptin is on, AMP kinase will be on. And as we have seen in the previous slide, if AMP kinase is on, ACC will be off, okay, because it will be phosphorylated. And if ACC is off, okay, there is not going to be any generation of malonyl-CoA for fatty acid synthesis. Malonyl-CoA will go down. If malonyl-CoA is down, it will trigger upregulation of the carnitine palmitoyl transferase, the one that is absolutely rate limiting for determining how fast fatty acids are brought into mitochondria to be broken down, fatty acid oxidation. So let's recapitulate just for a moment. Leptin is there, leptin is on, AMPK on, ACC off, malonyl-CoA down, but it tells the cell, bring fatty acids in the mitochondria and burn them. So leptin on, fatty acid breakdown. Okay. So that will be, so if this hormone is on, basically you lose all your fat or you burn your excess fat. Okay. Is that important? Well, when in doubt, knock it out. And what happens when leptin is not around to inhibit malonyl-CoA synthesis and fatty acid synthesis? This is what you get, the genetic model. Okay, you get an obese mouse. That's a normal mouse, and that's the knockout. And the effects are stunning, actually. So leptin has been used as a target for obesity. It has been studied extensively for obvious reasons. So now, if you can find drugs that can interact or block this interaction, you can see how you could potentially have an obesity therapeutic, and a therapeutic to fight obesity. So the take-home message are extremely important step in the regulation of fatty acid synthesis, heavily regulated. And if you can interfere with drugs and therapeutics, then you've gone a long way. All right, so we're going to stop at this point, and then we're going to discuss something about how some of the more complex lipids are made in the next video.